Hey everyone. I hope you all had a wonderful 4 days. I know I did. Weather was beautiful. And we are getting very, very close to the end. So that's exciting for everybody to not have school hanging over your head, at least for a couple months, unless of course you're doing summer school. So just a little rundown of what we're going to be doing. Today we are going to get into our special census part. I do have a quiz that's posted. Last I looked, which was early hours this morning, I think I had 4 people that had written it, which is fine. It closes at noon tomorrow, so make sure you're getting that done if you haven't done that yet. I did mark and get back to you the CNS PNS worksheet, other than the people that handed in late. So if you handed it in late, I haven't got to it yet today, okay? Couple other things, let's see. I do need to apologize to a couple of you. I didn't realize until this morning that I had put that we would be having a class yesterday, which was the 19th. Somehow I got the numbers confused in my head. I meant Wednesday, but I said 19th, so sorry about the confusion on that. We are almost done the nervous system, so we have one more booklet to do. So you'll have one worksheet that I will post as soon as class is over today. That goes up to the end of the eye, and then we'll do our next lecture to the end of the ear on Friday, and then you'll have a special census quiz next Monday, and then probably the end of the week, maybe on the Friday, I'll open up a unit test. And then we're just got reproduction and we're done. Okay, so because we have lots to do, let's get started. So we will get into the special census. I did share this with you earlier this morning. So let me get that opened up so we can see what we're doing with special census. All right, we'll just get to the beginning, like so. And like so. All right, so just a summary of what the outcomes are for this section here. Describe the structure and function of the parts of the eye. So we'll get into the eye at the end of class today. The cornea, lens, sclera, choroid, retina, rods of cones, foveus centralis, pupil, iris, and optic nerve. So those are all the things we need to know about the eye. Structure and function of the parts of the human ear, including pinna, auditory canal, tympanum, ossicles, cochlea, organ of Corti, auditory nerve, semicircular canals, and eustachian tube. Whew, lots of stuff. And then explain other ways that humans sense their environment and their spatial orientation in it. So a couple of suggestions there as well. Yeah, so let's, without further ado. So we're gonna just start talking about senses, okay? So first thing we need to talk when we talk about senses is sensory receptors. So basically the receptors are, and we've talked about these, right? We know that they have to detect things in our environment in order for us to perceive, right? Think about or understand what's going on. And we've talked about them as part of our reflex arc and that they send messages to the sensory neuron, okay? So what they do is they take a form of energy and then they convert that into electrochemical energy, i.e. leading to depolarization of a sensory neuron, right? They send a message through neurotransmitters to sensory neurons. Okay, so that's what our sensory receptors do for us. So I gave you an example here. Our light receptors take light energy and make it into electrochemical energy. Mechanical receptors take mechanical energy and make it into electrochemical energy. So those are the types of things. Chemoreceptors take chemical energy. So a few of the receptors that we're going to be talking about, one of them is a mechanoreceptor. So mechanoreceptor is stimulated by the cell becoming deformed in some way. Okay, so mechanoreceptors are stimulated by being stretched or being bent. And so when I talked about the receptors in the birth canal, so I said when the head moves through the birth canal, it causes stretch receptors to be activated. That sends a message to the hypothalamus, oxytocin gets released. Stretch receptors, so the cells are being stretched, right? So mechanoreceptors could be with blood pressure, right? We have baroreceptors that detect what our blood pressure are. They're being stretched, and that tells us that the pressure is getting to be high, right? So our mechanoreceptors, are for different parts of our body here. There we go. So sense of touch, right? So we're going to deform cells. Pressure, we talked about that. Vibration. Hearing. Ooh, weird. You think hearing is sound energy, but it's not. Hearing receptors are mechanoreceptors. What we have is hair cells in our inner ear, and we'll talk about this later. And basically, they have a little membrane above them, and those hair cells get displaced. Okay, there's fluid that moves, hair cells get displaced, they hit this membrane and they bend. And as soon as they bend, they depolarize, sending their message through a sensory neuron, which goes through the auditory nerve to, of course, our temporal lobe of our brain. Okay, equilibrium, that's our sense of balance. Weird that that's a mechanoreceptor, but it's the same thing. We have hair cells that bend, depolarize, sending a message eventually to our brain. Okay, we have thermoreceptors. So they're stimulated by changes in temperature. Now, one thing we need to remember about that, they're not temperature detectors. They detect change in temperature, right? Everything is relative. So we walk from a warm room into a room that is 2 degrees lower, it's going to feel like a cool room, right? But if we go from a room that is cold into the same room, but the room we started in was colder, maybe 10 degrees colder, the cool room is going to feel warmer. So we detect change in temperature, not temperature. It'd be cool if we could. We can't put our hand in something and say that's 62.7 degrees Celsius, right? But we can tell whether it's warmer or cooler than other liquid, okay? Photoreceptors, photo is light, so these guys are stimulated by light. They're found in the retina of our eyes. Chemoreceptors are detected or stimulated by the presence of chemicals. Chemicals sort of lock into the chemoreceptors like a lock and key mechanism. So we have these on our tongue, we have these in our nose, and we have these in our bodily fluid, or in parts of our body, right? Because our body can detect the presence of carbon dioxide, and when that gets to be too high, okay, and then it would cause our breathing rate to go up to get rid of it. Okay, sensory organs. These are all the organs that are responsible or that have sensory receptors and connective tissue with them. So these are the things that we learned about way back in kindergarten, when you were 5. Okay, 5 senses. Sensitive sight, taste, touch, hearing, and what did I miss? Smell. Okay. And what I like about this image is we see the part of the body and then we see where it's associated in the brain. So we see touch and taste is here in the parietal lobe, and we see sight is here in the occipital lobe, and smell and sound is here in the temporal lobe. Okay. So they're groups of receptors, because we have to remember that we don't just have our groups of receptors and connective tissue. That tissue helps for those receptors to work. But imagine our photoreceptors were just sort of sticking out, and then we fell down, and now we're blind, right? We have all this connective tissue that helps to protect it and make it work properly, right? Like our size of our pupil can change to allow or disallow light into our eye, so we don't damage the retina by having too much light exposure, okay? Sensation, to be aware of what's going on around us, right? So we talked about this a little bit. In order for us to perceive or understand that something is happening, right? So just because our sensory receptor detects something, it hasn't got to our brain yet. We talked about that in reference to reflexes, right? So just because our receptors detected a sharp object, right? Our brain doesn't detect sharp object yet, right? It takes time, and it has a pathway to get to. So for us to perceive the stimulus, first of all, the stimulus has to be capable of making a response in our body. So it has to meet a certain threshold level. Okay, so our stimulus can't be where it is below a threshold level. So for example, a frequency that's too high or too low, because we have a certain frequency that we can hear, which actually starts to decrease as you get older. So you guys can actually hear some higher frequency sounds than I can hear, just simply because you're younger than me. Okay. We need a receptor sense organ that can convert that into a nerve impulse, can take that energy that we've received and convert that into electrochemical energy. Okay, so if we don't have receptors, we're not going to perceive what's happened. Okay, we need a nerve to carry that message to the brain, right? So with our eye, it's the optic nerve. We have several different nerves in our skin that are going to carry that message to the brain. So it's got to get to the brain, and then excuse me, the brain has to translate that into something that we can perceive. So if we meet step 1 and step 2, when we have stimulus and let's say our eyes and our photoreceptors are working just fine, I'm still not going to perceive that image that my sensory receptors are detecting. If I have damage to my optic nerve, that message doesn't make it to my brain, I don't perceive it. Or if I have damage to my occipital lobe, then I can't process that message, I don't see it. Okay, so it has to be able to go through these pathways, so each of these steps, in order for me to be aware that that's there, to have an awareness of it. Okay, now one interesting thing that our bodies do is something called sensory adaptation, and basically it means that we lose our sensitivity to some kind of stimulus because we're constantly bombarded with it. And what's interesting is this happens with our brain. Our brain does this job, so it doesn't have anything to do with our threshold level of our neurons. But basically, this is a mechanism to help us focus on what's new and important in our world around us. Okay, so when I'm saying this, I'm saying that it's important for us our brain to know what's new, not what's already happened or what's old information. Okay, so this morning, I'm assuming that all of you woke up at some point. I'm hoping that you're not lying in bed, but some of you might be. We put on a shirt. Okay, so this morning you put on a shirt, and as you put on your shirt, it touched your skin, and as it touched your skin, your brain is aware of that, going, okay, skin's touched, you are wearing a shirt, right? But you go through your day, your brain knows that you're wearing a shirt, it doesn't give you that message anymore, right? So you're not sensitive to it anymore. So you don't think about it anymore. It's gone, okay? Because then we can focus on the new stuff, more important stuff that's happening. Other example, okay? We don't eat a lot of bacon in our house, but when we do, right, bacon has this very strong aroma, fills your house, okay? But after a while, it's gone, right? Your whole house has this odor of bacon, but after being exposed to it for a period of time, it's gone because it's old information. Okay, your brain says not new anymore, don't care. But then you go for a walk. Okay, you go for a walk for a couple hours, you come back, bacon, right? Because it's new, it's bacon, right? Or the air conditioner being on, or your furnace kicking in. When it first kicks in, oh, the furnace kicked in. And then you're doing stuff, you're not hearing the furnace going constantly. Okay? Could you imagine how it would be if you had to walk or drive to school or do a math problem or sit in class or what have you, and you're constantly getting this message, right? Bacon, 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 wearing a shirt, bacon, bacon, wearing a shirt, air conditioner's running. How could we ever focus on anything else, right? So this is our brain's way to allow us to focus on new, more important information, all right? So ability to adapt depends on the receptor. Pain receptors don't adapt, and that's really important to us. Now, we can stop feeling pain. It could be because we have endorphins that have kicked in, but they don't adapt. So if you have rolled your ankle, you're playing a sport, you rolled your ankle, right? You don't just sit for a while and your body's like, okay, not new information anymore. Go out and play some more soccer, right? That's not going to work because that is going to cause more damage to that body part, right? So pain is an important message to us. We don't want to lose sight of that. Okay, so that's what sensory adaptation is. We've all experienced it in some way, like the shirt you're wearing today, for example. Okay, so let's look at the skin. So sensations associated with our skin. So we have pain. We can sense cold. We can sense hot. We can sense light touch, stronger pressure touch, right? So all of those things associated with our skin. When we get those messages, what part of the brain do those messages end up in? Let's see how well you guys remember this. What part of the brain do those messages end up in when we get those messages from our sensory receptors in our skin? Thank you. The parietal lobe of the brain. Exactly right. The parietal lobe of the brain is where these messages are going to end up. Okay. So the receptors in the skin send a message to a sensory neuron, which is within a sensory nerve. Now, I've isolated or I've named a couple of nerves that you need to know. If I don't name them, you don't need to know them. So this, I'm not naming it. I'm just saying it's generically a sensory nerve. So you just need to know that. Okay, so the sensory nerves, the receptors in our skin would include mechanoreceptors, thermoreceptors, and then pain receptors, which are called nociceptors, but you don't have to know that. Okay. And the message here is processed in the parietal lobe of the brain. All right. Taste receptors. Okay, so these guys we know are on our tongue. That's where we taste things, although our sense of smell and our sense of taste actually do work together as a team. So we have chemoreceptors on the taste buds of our tongue. Okay, so here's the receptor cells we see in the bottom right corner here. And this is a taste pore. These are our food particles, and they're going to attach to these receptor cells within our taste buds. Now, there was a thought at one time that different parts of our tongue had taste buds for different types of tastes. That has since been debunked. That's been debunked probably for the last 10 years or so. Each of our taste buds has receptors for these different tastes. And the different tastes we have are sweet, salty, sour, bitter, which you've probably heard of before. And then there's another one called umami. And umami is a Japanese word for deliciousness. And it's kind of like that. To me, when I think umami, I think like stew. So it's sort of a meaty, savory flavor that we get from umami. Okay. All right, in order for us to taste something, the chemicals have to be dissolved. So our saliva would dissolve them. And if you wanted to test this at home, you could. You could have your sugar and salt, put them in bowls, close your eyes, figure out which one, like, so you don't know, well, then you wouldn't know which one you're using. Label them and then pick a bowl. And put a little bit on a dry tongue and you won't be able to taste anything. So sugar or salt looks the same, but until your saliva mixes with it. Okay, so chemoreceptors in the tongue send a message to a sensory neuron within a sensory nerve. I didn't give you the name of the nerves. You don't need to know it. Okay, processed by the parietal lobe of the brain. So again, we need to understand there's a pathway to the brain. From the receptor, it's going to go to a sensory neuron, which is within a particular nerve, which is going to make its way to a part of our brain. So we have the perception of it. Okay. Smell receptors, so these are chemoreceptors. For smell, they have a special name called olfactory cells, and they're found in our nasal membrane. And we can recognize cells like up to 10,000 different smells. Now, we require a higher concentration to be able to detect it, more so than a dog. A dog just needs a low concentration. And probably for us, this is to help us focus on more important things. So could you imagine walking through the mall and you're focused on, hey, Jim was here. Oh, I smell Susie. Susie must have been here a little while ago. Oh, this guy smells pretty bad. Bad as in like mean, right? we could get so caught up in all of those smells, and that's why dogs, when you take them for a walk, they want to smell everything, right? Where we lose all of that because we're focused on different things. Chemoreceptors, this should say in our nose. I was quickly doing this, sorry. Chemoreceptors in our nose send a message to the sensory neuron, which is within a sensory nerve. So again, we don't have to know the name of the nerve, which goes to the temporal lobe of the brain. Note to myself to fix this. All right. And here we see there's our cells, or sorry, our molecules. So there's the chemical, could be a perfume, could be body odor, doesn't matter. It's just some chemical, and it's attaching to specific receptor sites, okay? And here we see the receptor sites here. There's our chemoreceptor, and then that's carrying it up to our olfactory nerve, right? Here's our cells, so those are the sensory neurons, and then that'll make its way to the temporal lobe of our brain. This is the olfactory bulb. We have them sort of on the base of our frontal lobe. If we flip the brain over, that's where that olfactory bulb is. So the bulb's right here. Okay. All right, so those are our other senses. Now we're going to talk about the eye. Okay, so the first thing we need to do is talk about the parts of the eye that we need to know about. So let's start off at the very top here. Oh, did I lose my... Okay, sorry guys. Just did my formatting wrong with this, I think, but we'll just write it in for now. This is the sclera. Okay, so the sclera is the white of our eye. Okay, and it's a thick, fibrous part of our eye, helps to maintain the shape of our eye. Okay, and the next part, so our eye is basically 3 layers. So our first layer is that outer layer, that's the white part that we see there. The second layer is referred to as the choroid. And the choroid is a really dark pigment. That's where all our blood cells run through. So all of our blood vessels run through the choroid layer. And it's very dark, so it helps to absorb light. And then our innermost layer is the retina. This is the photoreceptor layer. So our photoreceptors are in that layer. It's really thin, kind of this creamy, transparent layer. Okay, so now let's just take a look over here. We'll talk about light entering the eye. Okay, so as light enters the eye, light's coming through here, the first part of our eyeball that it covers, or that it hits, oh, now I have sclera. Huh, I must have put these in a weird order. Didn't need to do that. Okay. The first part of our eye that it's going to hit is called maybe none of these are in the right order. There we go. It's called the cornea. Now the cornea, you can't really see it because it's clear, right? It's transparent, but it actually is an extension of the sclera. The sclera is white, the cornea is clear, but really they're the same thick fibrous material. Now behind the cornea is what's referred to as the aqueous humor. So it's this watery layer. So it's basically very, it's not very viscous. Okay, so it's very thin liquid here. And part of what it does is it helps to provide nutrients to the cornea, because there's no blood vessels here. If there was blood vessels in the cornea, we'd be blind because the light wouldn't be able to get through. So by diffusion, a lot of the nutrients are going to feed these cells in the cornea coming from the aqueous humor. Okay, so our light is traveling through the cornea. Now, those of you who have taken physics and talked about light, you know that every time light hits a new medium, it bends, and it bends, and it bends, and it bends. So light hits the cornea, it's a different medium than the air, so the light's going to bend. And then it hits the aqueous humor, that's a different medium than the air, so it's going to, or different medium than the cornea, so it's going to bend. Okay? Then light's going to travel through the pupil. Now the pupil is just a hole. It's just a hole, but remember we can't actually poke through the hole of our pupil because it's covered by the cornea, right? but it is simply a hole that allows light to get through, okay? So because it's a hole, it's not a new material, there's no difference in the refraction of the light. Next, we have the iris. So we see the iris is here and here. The iris is the pretty color of your eye. Whether you have green eyes or brown eyes or blue eyes, that's that color of the eye, that's your iris, and it's a muscle. And it's a muscle that controls how big your pupil is, okay? And if we take a look, we see this choroid layer. Well, we see how it's connected right here to the iris. They're actually the same layer of darkly pigmented material, okay? All right, then we have the lens, and the lens is this flexible material that can become flatter or rounder that allows us to focus on images, okay? And light has to pass through the lens, and when light passes through the lens, it's a different medium, so the light refracts, it bends, okay? Then the light keeps on traveling, okay? And actually, we have these ciliary muscles here, and they're part of the choroid layer, and they actually control whether the lens flattens or not. So they're a muscle. The lens itself is some fibrous material, but the ciliary muscle helps control whether it gets flatter or thicker, okay? All right, then the light has to travel through this space. So in the center of our eye, it's not just this big empty space, it's filled with this gel-like fluid called the vitreous humor. Because this is a different medium for light to go through than the lens, the light bends again, okay? So light hits the cornea, it bends, goes through the aqueous humor, it bends, goes through the lens, it bends, goes through the vitreous humor, it bends, and all of that bending is supposed to cause the light to hit right here. So our central vision, whatever your eyes are focused on, that light hits this spot right here called the fovea centralis. All right, we have a couple other spots here in our eye. The first area here that we have, this is the blind spot. And the blind spot is where our optic nerve attaches to the retina, okay? There's no photoreceptors there, so we actually have this gap. Now, it's in our peripheral vision. It's not in our central vision, because our central vision light goes here, but on our sides, we have this little gap, this space where we don't see anything. And we'll check out where that is in a bit. And then here's the optic nerve. What part of the brain does the optic nerve carry its messages to? What part of the brain does the optic nerve carry its messages to? Well, there's 17 of you out there in cyberspace. Someone's got to know the answer. What part of the brain does the optic nerve carry its messages to? Thank you, Grayson and Emily. Occipital lobe. Exactly right. Perfect. Okay. Now, let's keep moving here. Okay, so we're just going to look at all of these parts individually. So we have the sclera, which is the outermost layer of the eye, the white of the eye, otherwise known as. Okay, it's tough connective tissue, and its job is really to maintain the shape of the eye. Okay, so it helps. So when we fall on our face, our eye just doesn't go and suck in, right? It's this nice tough material. It helps to protect the inner parts of our eye. And it's really tough. And if we were doing the dissection, you'd see that, because you really have to saw at it to open it up. Just gently saw. And it is filled with fluid. So over the years, I have had students spray themselves with vitreous fluid. Okay. The cornea, so it's a continuation of the sclera in the front of the eye. Remember, it is clear. Okay. It's transparent, light can get through it, so it doesn't have blood vessels, because if it did, we'd have a problem, we'd be blind. So its job is to bend light towards the pupil. And then the aqueous humor is the watery fluid here, right behind the cornea, that's going to provide nutrients to the cornea, because the cornea is not in connection with blood vessels, so it needs those nutrients from different means. Okay. All right, the lens. My most favorite part of doing the dissection is touching the lens. So the lens feels very, very hard when we're dissecting it out of a dead eye. Our eye, it's going to have a lot more flexibility, yours more so than mine because you're younger than me. But it does look like this. So it says biconvex, so it's rounded on both sides, and light goes through it, and it can flatten and narrow depending on whether we're looking at something far away or close up. Okay. So the ciliary muscles allow for that to happen. What else was I going to say about the lens? The lens is a crystalline structure, and I haven't been able to do this for the last couple of years, but if you have a nice clear lens when you're dissecting, you can actually put it on a piece of newspaper, and it's like a magnifying glass. It actually magnifies things a little bit more than what they were. It changes shape to focus images. Ciliary muscles are the muscles responsible for controlling the shape of the lens. Okay, so we can see that here. And again, we want to notice the focal point of the light is right here at this point. Okay, so that's called the fovea centralis. Then the light has to travel here through the vitreous humor, and that's the gel-like material that fills the space. So it's not this big empty space with our eye. Okay. And part of the purpose of that is the same as what our sclera is to maintain the shape of our eye. So again, if we fall, if we didn't have that gel in there, our eye could suck together, and we have this strong connective tissue that's not going to pop, right, the second that we fall. So these both help to maintain our eye and therefore our eyesight. The choroid is our middle layer of our eye. Right? So we have the sclera, and then behind that is the choroid. And what's neat with the eye dissections, you can actually peel these layers back and see an individual layer. Okay. So the middle layer of the eye is the choroid. This is the part that's got the blood vessels in it. It's darkly pigmented, so it can absorb light. And then we have the iris, and again, we can see how the iris is an extension of the choroid. Okay. So when we see the underside of the iris, the side that's closest to the lens, it's very dark, right? That dark pigmentation. And the iris, we want to remember, is a muscle. It's not one we have voluntary control over, but it is a muscle that controls the size of our pupil. Okay. The pupil is the structure that allows light into our eye. Okay, regulation of pupil size. So our iris is in charge of our pupil size. Okay, so the constriction of our pupil will constrict when we're exposed to bright light. Okay, so when we're exposed to bright light, our pupil constricts. So this says the iris relaxes. Remember, that's the muscle. It's relaxing in, okay, and it's contracting out. Relaxing in, contracting out. So the iris is contracting, so the pupil dilates. Okay, the pupil's getting bigger when we're in dim light to allow us to see more light. Okay. Our pupil also constricts when we see or looking for something close up. Now, one thing I usually do, it's a little harder this way, but if I'm taking a look at something, right, and you can try this, is take it and put it close to your face until it becomes blurry, whatever you're looking at, a piece of writing or something. And then, at that point, take your hand and make a little crook so you have a little tiny hole in that crook that's about 2 millimeters by 2 millimeters, and then try to read. And one letter at a time, you should actually be able to read it because we have this constriction of light. So we can focus on a tiny bit. It's not very efficient, but we can actually see one letter at a time as opposed to not being able to see it at all. So the first glasses that were made use this function. So they had these little pricks in bone, and then you could see little tiny bits at a time, but it actually focused that image. Our pupil dilates when we're in dim light. When we're looking at something far away, and then also as part of our fight or flight response. Okay. Our retina is the innermost layer of our eye, and it has photoreceptors. Okay, so we have two types of photoreceptors, rods and cones. So we see the rods here, and then we see the cones here. We actually have three different kinds of cones: blue cones, green cones, and red cones. And then how many of each are stimulated allow us to see our different colors. So it's just like an RGB cable, right? Red, green, blue. So as much whatever stimulated group in that particular grouping, whatever you're focused on, that'll indicate what color we see. Now we have about 20 times more rods than we do cones, okay? Rods are responsible for responding to low intensity light. So the light right now that I'm exposed to with the light coming in from outside is high intensity light. Low intensity light is like the light that's in your bedroom when your lights are all shut off at night, right? So you have the street light coming in, so you can still see the shapes of things in your room, but you can't see all of those colors in your room, okay? So cones respond to high intensity light, and cones allow us to identify color. Rods are low intensity light, we can't see color. So in your room at light, when you just have the street light coming in at nighttime, you can't see the color of all the clothes that are sitting on your floor or the blankets that are on your bed. They're different shades of gray. However, rods do need some light. If we don't have any light, we don't see. And it's very rare to experience a situation where there's no light at all. We've done a couple adventures where we've gone for tours through caves or through coal mines, and if you get to a point where the light is no longer coming into the mine or the cave, and then you shut off all your light sources, you can't see your hand in front of your face because there is no light, right? We need to depolarize the rods for us to see, so you need some light to depolarize them. The cones have a higher threshold level, so they need more light in which to become depolarized. So when we think about rods and cones, sometimes we've had situations where, or you've experienced situations where you're lying in bed and you're kind of staring at your ceiling, it's dark in your room, okay? And your parents are getting frustrated with you because you haven't gotten up yet, and they come in your room and they flick on the light. And all of a sudden your eyes are in a lot of pain, the light gets flicked on, but what's happening there is every rod is all suddenly depolarized, and that causes a momentary amount of pain until the rods will start to, they've all depolarized and now you don't feel that anymore. Another thing that we've all experienced is when you go from a bright area, so maybe it's bright in your hallway, to your dark room, and you don't feel like turning the light on, it takes a while for your eyes to adjust to the dark. Well, what's happened is all of your rods have depolarized in the hallway where the light was, and then coming into your room, they have to recover, and then only the ones that are being stimulated by the light in your room will depolarize, and then that allows you to adjust to that low intensity light, okay? All right, the fovea centralis, so remember I told you that's where the light hits, right here. So when you have your central vision and you're looking at something, the light hits right at the fovea centralis. That is the center back of our eye, okay? That's where the cones are. So the cones allow me to detect light or detect my images, detect color. That's where cones are and no rods, okay? But as we move here into our peripheral vision, so that's the vision I have here and here, that's where the rods are. So our peripheral vision should be in black and white, except our brain tries to fix that for us and add some color, okay? So that's our peripheral vision, and we depend on that a lot. Like if you're playing a sport, you depend on that to kind of feel where or see where everyone is around you, right? We depend on that for driving, is our peripheral vision. Okay, so that's where our rods are, is in our periphery. So our blind spot is in our periphery as well, where the rods are. Our cones are right in our central vision. Okay, so the fovea centralis is when we're staring straight at something ahead, that's our vision. As soon as we move, the image that's on our fovea, right, changes because I'm focusing on something different, which means different cones are being stimulated to allow me to see the different colors. Okay, my blind spot, so that my blind spot is right here, and that's where the optic nerve is attached to the retina. We don't have rods or cones in our blind spot, right? What I like about this image is we can see here, right? This is our fovea centralis, so the center of the fovea, fovea centralis. So the red here is my cone, so I see lots and lots of cones there. And then here, I see nothing, right? There's no photoreceptors there. So any light that lands in this spot, we don't see any photoreceptor, or we don't see anything because there's no photoreceptors there. Okay. All right, our ciliary muscle is a muscle that's attached to our lens. It's actually attached to ligaments that are attached to our lens that alters the shape of our lens. Okay. Accommodation reflexes are reflexes that allow us to see things that are far away or close up. So when I'm trying to focus on something that's far away, my lens flattens out. Okay, so that's what I'm seeing here. As I'm looking at something that's far away, my lens has to be flatter in order to focus that image right on the fovea centralis. Okay. The other thing is my pupil dilates. So those are the two things that happen in order for me to see something far away. For me to see something close up, my lens thickens. Okay, because again, the whole point is to get that image on my fovea centralis. So the lens thickens and my pupils constrict. So those are the two accommodations that are made to see something far and the two accommodations that are made to see something close up. Let me just, okay, let's do this last little bit. Okay, aging. When we get older, so I am older. Your parents are older than you. When you get older, what happens is we end up with this protein that sits on top of our lens, and our lens starts to lose its flexibility. Okay? So lens, remember when we want to see something far away, our lens flattens. When I want to see something close up, our lens rounds, right? Kind of think of your lens as a rubber band, right? So the rubber band gets pulled flat, and then that energy drops off the lens and it bounces back to round. Hold flat, bounces back to round, okay? Just like an elastic band. Well, when you age, those proteins, it goes like this, it flattens to see something far, and then your lens is like, creak, and it really doesn't bounce back anymore. So it stays kind of flat, which means that as you get older, you see things far away better than you see things close up. So you might notice that your parents start to do this. They start to extend their arm when they want to read something. So little kids have a very, they're nearsighted by, that's just how they are. They're nearsighted, which means to comfortably read something, they need it to be close to their face. It's about like 7 centimeters and then 10 centimeters. And then all of a sudden, somewhere between 40 and 50, that point, that accommodation point, jumps to about 50 centimeters. And it starts to get a little longer than your arms, and it starts to be where it's really hard to read something that, because too close is your eyes don't focus anymore. Okay, so it has a fancy name, presbyopia. Don't have to know that name, but basically what we're seeing here is our lens isn't rounding enough in order to see our image. So the focal point of the image actually ends up being behind our eye, right? So we don't get this nice crisp image. So this just shows the corrective lens that you would have. So I have a corrective lens. I have progressive lens in order to correct for that image. Okay. All right. That was very, very fast. We didn't watch any of the videos or do the blind spot test, but we will do that next class. And then I'll talk a little bit about what a dissection would have looked like next class as well. The worksheet I will share with you in just a couple of minutes. So you'll have that. And yeah, and then we'll talk about a couple of little eye things, watch a couple eye videos, and then do the ear next class. And then we're done with special senses. Any questions? All right. Well, you guys are awesome. So we will see you on Friday. And the sun's trying to shine, so maybe dip yourself outside a little bit and get a little bit of vitamin D. And that's it for today. So you guys have yourselves a wonderful rest of the day, and we will chat with you on Friday. Eric, sorry, I just saw your question here. Would LASIK prove ineffective if you have a faulty lens? If your lens was faulty, maybe, but they could accommodate with your cornea to accommodate for the lens being faulty. So yeah, because the LASIKs, it doesn't, you can have a mono lens, which just gives you one level where it doesn't flatten or thicken, which you'd still have to wear glasses for. But for the LASIK, yeah, the LASIK is just the cornea. And I believe that they can accommodate and adjust your cornea to make up for flaws in your lens to some degree. If the lens isn't transparent, then no, then you have to get a lens replacement. I hope that answers your question. And yeah, that's it. Any other questions? All right, guys, have a wonderful rest of your Wednesday.